Hello everyone, I am Rajeshwari Moganty, Professor at the Department of Biochemistry, All India Institute of Medical Sciences, New Delhi. Now this module what we are going to today is number 29 in the paper on membrane biophysics paper number 12. Now the topic we are going to do today is on the membrane current and propagation of the nerve impulse and synaptic transmission. So this is actually in continuation of what we did earlier. We learnt about the action potential, resting potential, action potential, refractory period and various stages in terms of the current and how the ion the potassium and sodium ions change their concentration. Okay. Now in continuation of this, I would again like to tell you that all animal cells are actually surrounded by a membrane compound, that is a lipid bilayer, which you did it in any cell membrane in the initial lipid membrane, which you did in one of the initial modules of this paper. Now the ion transporters or the pumps of the proteins actively push the ions across the membrane and establish concentration gradients. Therefore, the ion channels allowed to move across membrane and the concentration gradients actually help, therefore, create a voltage between the two sides of the membrane. The membrane potential is a difference in the electrical potential between the interior and exterior of the biological cell. Now, with respect to the exterior of the cell and in typical values of the membrane potential is generally between -40 to -80 millivolts. So, what is membrane current? The membrane current, the electrical signals within the biological organisms are actually in general are driven by ion-ion interactions across the cell membrane under the influences of for example, diffusion and electrical fields. Now, the ion pump influences the action potential only by establishing the relative ratio of the intracellular and extracellular ion concentration. For example, if you take the sodium potassium pump contribution to the membrane potential. So, the pump has to have three effects. One is that it makes the sodium concentration high in the extracellular space and low in the intracellular space. And the second one, the membrane the pump actually creates or makes the potassium concentration high in the intracellular space and in the intracellular than compared to that low in the intracellular low in the extracellular space. Okay. So, the pump has actually three effects. What are they? The one is it makes the sodium concentration high in the extracellular space and low in the intracellular space, whereas the second one, it makes the reversal, that is potassium concentration high in the intracellular space and low in the extracellular space. Now, what is the third one? The third one, the pump actually gives the intracellular space a negative voltage with respect to the extracellular space. So, these are the three main functions the membrane membrane pump actually creates. Now, the ion pumps play a significant role in the in case of the repolarization of the membrane after the action potential occurs. So, this figure actually shows you how the channels open up as compared to, as there is a signal and then the ion come and the channels open up and then again they close as far as the extracellular space and intracellular space is concerned. The ion channels are actually integral membrane proteins and they have a pore to pass the ions and with a pore through which ions can actually travel between the extracellular space and intracellular with the cell interior, they pass through these ion pumps. The action potential actually involves mainly in the opening and closing of the ion channels, not in the ion pump. The second point is that when a channel is open, ions are permeate through the channel pore down the transmembrane concentration gradient for that particular ion. The third one is that ionic concentration differentiates The differences in these ionic concentrations, ion channels and membrane capacitance, they contribute to the electrical properties of the membrane. So, the membrane capacitance is actually determined by the properties of the lipid bilayer, across which the concentration changes do happen. The voltage of each ionic pathway is actually determined by the concentration of the ion on each side of the membrane. Now, the last point is that the conductance of each ionic pathway at any time, any point of time t, is actually determined by the states of the all ion channels, and that actually potentially permeable to that particular ion. Therefore, including the leakage of the channels and ligand gated channels and voltage gated channels. So, based on this little bit of introduction to about the membrane, role of the membrane in the nerve impulse transmission, the examples that this actually leads with the nerve impulse is a sum total of physical and chemical disturbances created by the stimulus, whether it is electrical or chemical or mechanical, in a neuron or a neuron fiber. Now, this results in the movement of the wave along the nerve fiber. Therefore, this module actually deals with membrane current, propagation of the nerve impulse, synapses and synaptic transmission. Now, based on this, we will do first the propagation of the nerve impulse, then the rate of the conduction of the nerve impulse, synapse and synaptic transmission, and finally, of course, I will summarize what we learnt in this. Now, the action potential actually has been explained to you earlier, but nevertheless, because of the need in understanding this particular topic, I would like to tell you that action potential is actually an action. The membrane current flows out in 4 different phases. That is, firstly, the outward current results from the brief 200 microseconds of the stimulus of the current, which is shown in the next figure to you, and most of this current actually flows through the membrane's capacity rather than through any channels. That is one thing important. The second phase is the large surge of the outward current which reflects the very high rate of depolarization, which is in the order of 1000 volts per second, and during this rising phase of the action potential, the inward sodium current supplies this outward capacitive current. The fourth point, that is, as the action potential approaches its peak, the capacitive current reverses polarity and approaches 0. The capacitive current crosses 0 precisely when the action potential reaches its peak, that is the threshold, because at this moment the rate of change of the voltage is 0. Now, throughout this, the long falling phase of the spike, the capacitive current is inward and now of a small amplitude. This smaller amplitude, you see at the end of the peak, you see that the phase is actually small in amplitude. This smaller amplitude reflects the much lower repolarization during the fall of the phase of the action potential due to the competition between sodium and potassium currents. Next, slide 12 shows you the graph between time versus the millivolts voltage during the capacitive current period, stimulus period, as well as the where it is the flowing. So, they represent the membrane capacitive current flowing onto the membrane during the action potential evoked by a very brief depolarizing pulse. Now, let us see how this is actually potential difference actually propagates through the nerve impulse. The propagation of the nerve impulse actually occurs in 2 main parts. One is the origin or the stimulation of the nerve impulse, and then of course, later on the impulse is transmitted into the depolarization or the propagation or the travelling of the nerve impulse. So, first is the origin stimulation, then second is the propagation. Now, when it is originated, the first one, let us see how it is originated due because of the stimulation or at the site of the nerve impulse. In resting nerve cell, the surface is positively charged and interior is negatively charged. When the surface is stimulated, positive and negative ions are accumulated along the outer sodium and inner potassium surface of the cell membrane. Therefore, development of local excitatory state and development of the action potential occurs, and on stimulus sodium rushes inside cell, making the inner surface positively charged. The amount of sodium is not sufficient to generate action potential and returns to the outside immediately, causing a closed circuit, and the graph shows you the structure of a typical neuron and direction of the nerve impulse. Resting potential, in the resting state, the nerve fiber remains in polarized state and lies within minus 70 millivolts. The state of the depolarization, this is a tremendous increase in sodium conductance during the period, is known as the activation of the membrane, and after an initial slow rise, the depolarization waves overshoots rapidly and reaches approximately 30 plus 35 millivolts. The repolarization state, where reaches the isopotential line, that is potassium begins to come out from the inside of the membrane, causing outside the positive again. This is the repolarization. Then lastly, the spike potential, the periodic rise of the depolarization wave and rapid fall of repolarization wave are also known as the spike potentials. Now, this effect we have, this part of it we understood in a previous module, but here it is brought in again in a different context to show you how the polarization of the neurons membrane occurs along the nodes of the Ranvier and myelin sheath. These are shown here. So, the negative after potential, what happens? The approximately 2/3 of the polarization, the rate of the fall of being abruptly slowed down, and this slower fall is known as a negative after potential. Now, positive after potential is with the disappearance of the negative after potential, although rising of the membrane potential is achieved, at the resting ion status is not established. Therefore, it is achieved by the active sodium pump mechanism, which causes the positive after potential. At the same time, the potassium travels back into the inside of the membrane. So, this diagram 4 shows you clearly the depolarized membrane and the membrane at the resting potential. In both you can see that the inner and outer ion charges are different inside and outside the exterior to the membrane. Now, the propagation or also called the travelling of the nerve impulse. The propagation of the non-mediated modulated nerve fiber, that is when the fiber is excited at a point, the polarity is reversed due to the increased permeability of the sodium to the membrane, which develops the depolarization wave. Now, the positive current flows inward through depolarized membrane and outward through the resting membrane. Now, the depolarization wave travels in all directions along the entire length of the nerve fiber. The propagation, the second part is a propagation in the myelinated nerve fiber, that is a statutory conduction. In what happens in the myelin sheath is an active insulator, and therefore, ions cannot pass along this sheath, but the nodes of Ranvier actually permeate ions through it much more easily. Therefore, the impulse is transmitted from node to node rather than continuously along the entire fiber. There is no continuity, but only from the node to node. This jumping or leaping of the depolarization, this is a very important point, jumping or leaping of the depolarization from node to node is also known as a salutatory conduction. Slide 17, this is what is explained in this, that the propagation of the nerve impulse in this diagram has been shown non-modulated nerve fibers on a non-modulated nerve fiber, that is the axon is shown that in the action potential, then some part of the axon, how is the sodium is changing, then you have the sodium concentration changing, and then the potassium moving in. The rate of the conduction of the nerve impulse. Now, after it is started the propagating through the nerve, how does it conduct and what is its rate? The salutatory mechanism of the conduction in myelinated nerve fiber is very high and increases the velocity of conduction more than 500 times as compared to the non-myelinated or the non-modulated. Therefore, the rate of conduction of a nerve impulse actually increases with an increase in the cross-sectional diameter of the neuron and also the increasing thickness of the myelin sheath. These are the two that actually contribute to the rate of conduction of the nerve impulse. Now, the rate of transmission of a given neuron is a constant, and nerve impulse have a domino effect. That is, what happens, the receives an impulse and passes it on to the next neuron. So, the entire impulse actually passes through a neuron in about say 7 milliseconds by following the steps, which is shown in the previous one, that the total action actually finishes by about 7 milliseconds. The polarization of the neuron membrane and sodium on the outside and the potassium inside, when the neuron is not stimulated, the electrical charges are actually reversal. Now, then we will come to the phase where synapse. What is synapse? Synapse are actually junctional complexes between presynaptic membranes and postsynaptic membranes, which are actually the receptor surfaces of the recipient of the neurons, or where we also call it as effectors. So, the pre and post actually reply as it is described in this thing, that before the synapse and after the synaptic transmission, and the postsynaptic is the receiving end, that is a dendrites, soma and effectors. The synaptic knobs contain many membrane bounded synaptic vessels. They comprise of the neurotransmitters along the mitochondria, microtubules and other organelles. So, the synapses are named according to their location on the postsynaptic neurons. Now, that is why first one is the axiospinous the synapses, which are synapses occurring on the dendritic spines, and axiodendritic synapses are on the shafts of the dendrites. The third one is axosomatic synapses, which are on the soma of the neurons, and axonal synapses, which are on the synapses on the synaptic knobs. So, this kind of synapses, different types of synapses can occur. Synapses on the skeletal cells actually are, we call them as the neuromuscular junctions. So, once the synapse is there formed, how does a synapse transmit? That is a synaptic transmission. Synaptic transmission is actually a process whereby one neuron communicates with the other neurons or effectors, which are such as muscle cell or at any synapse. So, a typical neuron has a cell body, which I explained to you earlier, soma, and the branching is specialized to receive the signal, that is the dendrites, and a single process, that is the axon. The different parts are already shown in the previous slides. They carry electric signals away from the neuron and towards the neurons of other neurons, or the also called the effectors. So, the electrical signals carried by axons are action potentials. Axons have thousands of termini, branches ending as the synaptic knobs or synaptic termini. Now, at the synaptic knob, the action potential is converted into a chemical message, which is in turn interacts with the recipient neuron. So, so far we are talking about the signal and electrical signal. Now, it will be converted into a chemical message. The action potentials arriving at synaptic knobs trigger the release of neurotransmitter into a synaptic cleft. This slide shows you the structure depicting the typical chemical synapse and major elements in the chemical synaptic transmission, that is electrical to how the chemical signal is being converted. The synaptic vesicles actually fuse with the presynaptic membrane during this process of the exocytosis. These membranes of the old vesicles become part of the presynaptic membrane, and new vesicles pinch from the adjacent area of the membrane. These new vesicles are subsequently refilled with the newly synthesized or the recycled neurotransmitters. The released neurotransmitters diffuse across the narrow synaptic cleft. At the postsynaptic membrane, neurotransmitter molecules bind to the membrane bound receptor molecules with the recognition site specific for the neurotransmitter. Each one has its own binding site on the membrane. Binding of the neurotransmitter to the receptor, to its cognitive receptor, triggers a postsynaptic response specific for that receptor, and that can either be excitatory or inhibitory, depending on the properties of the receptor. If the receptor stimulation results in postsynaptic membrane becoming more electrically positive or the depolarized, then it is an excitatory postsynaptic potential, or we call it as EPSP. If the electric signal becomes negative, then that is hyperpolarized, then it is an inhibitory postsynaptic potential or IPSP. The excitation and inhibition depends on the properties of the receptor and not on the transmitter. Receptors can be coupled to sodium or calcium channels and all that are excitatory and produce a depolarization of the postsynaptic membrane, whereas receptors coupled to chloride or potassium anoles are usually inhibitory and they produce hyperpolarization of the postsynaptic membrane. Therefore, such receptors coupled to ion channels are called the ionotropic receptors, and those which are coupled are called coupled to the second messenger in the cell signaling process. Then systems that initiate a series of biochemical reactions in the postsynaptic cells, which are called the metabotropic receptors. Now, the metabotropic receptors can produce a different type of postsynaptic events, ranging from, say for example, the direct activation of the adjacent ion channels to alteration of the receptor membranes and sensitivity to the transmission of the transcription of the specific messenger channels, and like for example, the specific messenger ribonucleic acids, or even the activation of the specific genes that can also happen. So, once released into the synaptic cell, the neurotransmitters remain active until they are actually either altered chemically or taken back into the, that is the recirculated reuptake is taken into the synaptic knob by special carrier proteins or carrier systems, and then of course they are replaced, recycled. That means either it is altered chemically, substance chemical, or it can be recycled or reuptake will take place. Therefore, at cholinergic synapses, acetylcholinesterase is present in the synaptic cleft, and this enzyme, this is the most important one, is enzyme cleaves the neurotransmitter into acetate and chloride ion, neither of which is actually active. Serotonin and epinephrine on the other hand are taken up into the presynaptic termini and whenever they are recycled. So, I would like to summarize what we learnt in this module. So, in this module what we learnt is that ionic concentration differences between intra and extracellular ions, ion channels, membrane capacitance, which contribute to the electrical properties of the membrane, that is the membrane electrical properties. Then we studied the propagation of the nerve impulse, which involves the origin and stimulation of the nerve impulse, and of course, then the propagation and travelling of the nerve impulse. The rate of conduction we studied, this is actually increases with the increase in the cross section of the diameter of the neuron and also increases with the thickness of the myelin sheet. The next is the synapse. Synapses, we studied that they are the junctional complexes between the presynaptic membranes and postsynaptic membranes, and the action potential arriving at the synaptic knob actually triggers the release of the neurotransmitters into the specific, that is synaptic cleft, and hence the synaptic transmission takes place. Now, in the next module we will be studying more on the neurotransmitters, which are chemical messengers, and how they bring into the transmission takes place in further detail. Thank you.